Okay, continuing with the formed elements, the first of which is the erythrocyte, the red blood cell, erythrocyte cell. These are donut-shaped, flexible, small formed elements. They really have no nucleus, they have no cellular organelles, so there's no mitochondria, there's nothing. There's really just a cell membrane and filled with hemoglobin. They're a bag of hemoglobin. Now, the idea behind them is that their job is to transport oxygen. There's no organelles because they really don't have any metabolism. You wouldn't use a dog to deliver meat. You don't want any mitochondria to use up the oxygen that you're delivering. So they are just a bag of hemoglobin. Now, they are biconcave discs for a couple of reasons. The thing is that the way that the oxygen gets in and out of them is through diffusion. And diffusion depends on surface area to volume. So if these were like balloon shaped, if they were just spheres, then what would happen would be that the hemoglobin in the center would be further away from the outside, from the cell membrane, than other hemoglobin closer to the surface. So we minimize that by actually bringing the surface towards the center, by basically pinching it into this donut shape. By doing that, we increase the amount of surface area per volume and therefore facilitate the diffusion of gases in and out of the cell. So that shape is very important for the diffusion part. The other thing that this biconcave shape does is it makes them really slippery. They transport easily through the blood vessels. Think of what a snow tube looks like. If you go snow tubing at Snow Valley, what does the snow tube look like? It looks like a big donut. And the reason is because then that way you go very quickly down the run. You can bounce off the sides. You can bounce off each other. And you can flip over and go just as fast on the other side. It makes for less friction moving through the blood vessels, because literally these things have to go through the entire circulatory system in a minute. They inside there is some proteins called spectrum, and they provide the flexibility. It's what makes them bounce and not get hung up on each other. And as the cell gets old, the spectrum starts to break down and they become less flexible and less bouncy, and that's what limits their age. That spectrum can't be replaced because there's no nucleus and there's no ribosomes, there's no organelles to replace it, so the cell just becomes useless. Now, the size of a red blood cell is about 7.5 microns. The size of most capillaries in the body is about 7 microns. So that means that they have to fold ever so slightly to get through. But they are actually, especially in the spleen, at the very least, in most normal capillaries, they are going through in single file. And that, again, facilitates diffusion of oxygen out and carbon dioxide into them in the tissues. The opposite in the lungs. But they go through in single file, one after another. So when we look at it, about 7.5 microns, this kind of shape, this like a snow tube, like a donut. The photomicrograph is them going through a capillary in basically in single file, because they are about the same diameter as the capillary. Inside them, they have hemoglobin. The hemoglobin transports oxygen and carbon dioxide. When they are maximally loaded with oxygen, so each hemoglobin molecule having 4 oxygens attached to it, it's called oxygenated. When you lose even one oxygen from the hemoglobin, it's called deoxygenated. There is some globin, which is a protein with a tertiary structure. I don't really care that there's alpha chains and beta chains. What matters is in the center there is a pigment, and the pigment is called heme. At the center of each heme is an iron ion, Fe3+. And the iron binds to oxygen. So there are 4 heme groups, one for each of the globulin chains, and therefore each hemoglobin has got 4 hemes and then can bind to 4 oxygen molecules. The binding is fairly weak. It is reversible. When the oxygen binds, it can rapidly attach and rapidly detach. And what drives that is the concentration. So when the hemoglobin is in a high oxygen environment, it moves to the attachment side. When it's in a low oxygen environment, it moves to the detaching side. So it very readily moves in the direction to find an equilibrium. Now, the carbon dioxide binds very weakly not to the heme in the hemoglobin, but to the globin, to the protein itself. And again, it follows the concentration gradients. In a high carbon dioxide environment, it binds. In a low carbon dioxide environment, it detaches and is released. This is what the heme looks like. Please do not bother to memorize the structure. All that matters is there's 4 hemes, and each one of them has an iron ion, Fe2+, and that binds with oxygen. Erythropoietin, I've already said, is produced by the kidneys, and it's often called EPO. Now, people with kidney disease have to take injections of EPO because one of the things about having kidney disease is you don't produce enough erythropoietin, and therefore probably will have anemia. You won't make enough red blood cells because you don't have enough erythropoietin. The kidneys, primarily, react to a decrease in blood oxygen levels by making erythropoietin. And then the erythropoietin stimulates the myeloid cells to make erythrocytes and release them into circulation. The erythrocytes increase the oxygen carrying capacity. The increased oxygen in the blood then inhibits EPO release as a negative feedback, and the lack of erythropoietin inhibits making any more red blood cells. Testosterone enhances this, so it happens a little faster, so men tend to have higher erythrocyte counts, whatever. Anything that affects the amount of oxygen, therefore, affects the amount of EPO. Any low oxygen levels, so high altitude, for instance, will stimulate EPO production, and cause you to make more red blood cells. Any breathing difficulties do that. Your spouse being pissed off and holding a pillow over your face will increase EPO levels. It's anything that causes it. A loss of blood, so a desanguination, blood loss, causes less oxygen carrying capacity, which then stimulates the production of EPO, which makes you make more blood to replace that which you've lost. Anything that causes low blood oxygen levels. And so this is just a chart. Erythrocytes, because they have no nucleus and no organelles, their proteins only last so long and they can't repair themselves. So it takes about 120 days for them to become pretty much non-functional. So what happens is that the blood passes through primarily the spleen, but to a lesser extent the liver. It passes through the liver a lot, but getting rid of red blood cells happens mostly in the spleen, to a lesser extent in the liver. So what happens is the capillaries in the spleen are slightly smaller than the red blood cell. A brand new healthy red blood cell with lots of good spectrum is able to fold and pass through that capillary quite easily. If it's an old red blood cell, it's getting to be that 120 days old, then the spectrum is broken down and the cell isn't able to fold. So it hits these small capillaries and then can't get through them. They get stuck. And then macrophages in the spleen engulf them and break them down. It's like if you want to keep old people out of your club, make the doorway really low, and then if the old person is so stiff they can't duck down to crawl through the door, then you have the bouncers beat them up outside the door and drag them away. Same deal. So what happens is the phagocytes break them down. It takes the globin and breaks it into the amino acids and releases the amino acids into circulation where they can get used to make other proteins elsewhere in the body. The iron from the heme is made into something called transferrin, and then so transferrin, Fe is iron, so it transfers iron to the liver. In the liver, the iron goes to storage proteins, ferritin and something called hemosiderin, and it gets stored in the liver, a little bit in the spleen. As the red bone marrow needs iron for more erythrocytes, it gets transported to the red bone marrow, but just as needed. So the liver stores this ferritin. So it gets recycled. The amino acids get recycled. What doesn't get recycled is the pigment, the heme group without the iron. It becomes something called biliverdin. Verd means green, so it's the green shit in bile. It's the leftover red blood cells. In the liver, it becomes something called bilirubin. And then it's excreted. It's gotten rid of it in the body. It's a yellow pigment, and it gets mixed with the bile and put out of the body into the duodenum along with the bile. Bile is useful in the digestion of fats, but it also excretes this pigment. So most of it goes out that way. It oxidizes and becomes cercobilin, which is really a brown pigment that makes your poop brown. Some of it ends up in your blood and becomes urobilin and ends up being excreted out of the kidneys as a yellow pigment, which is why your pee is yellow. If you have liver problems and you can't make this happen, then the urobilin builds up and starts to stain all kinds of tissues, so the whites of the eyes and all kinds of tissues, and you start to turn yellow, and that's what jaundice is. And it's all the pigments from old red blood cells. We have millions upon millions of red blood cells per drop of blood, per milliliter of blood, and it's constantly being changed over. So we're making millions of them per second, and we're taking away. And most people, the production levels and the destruction levels are about the same. They're balanced. If you start to work out, the production starts to increase over the destruction. If you're going the other way, it will probably mean that you have excess, and and destruction will overcome production. With babies, they get their oxygen through the placenta, which is not as efficient as the oxygen that it may not be as efficient as oxygen that comes from the lungs. When the baby, therefore, is in the womb, the red blood cell count is fairly high to make up for this efficiency. When the baby's born, most babies, therefore, have too many red blood cells, because when they start breathing, their oxygen availability becomes greater than when they were in the womb. That extra red blood cells mean then that they have to be gotten rid of, because there's worries about viscosity. The body has to maintain a right viscosity, and so there's more destruction. There's therefore more bilirubin made, etc. There's less bile production, and so the urobilin can build up, and babies can become quite jaundiced until that balance is reached. Luckily, they're very small beings, and they don't have a lot of volume of blood, so it normally doesn't take very long. Some babies, it's not even noticeable. Others get a little bit of jaundice. So the red blood cells are made in the bone marrow. They pass through the holes in the capillaries of the bone marrow into the blood. They circulate in the blood for about 120 days. When they can no longer bend to get through the spleen and parts of the liver, macrophages eat them. When the macrophages eat them and phagocytize them, they break up the proteins of the globin into amino acids, which are recirculated back into the blood. They transfer the iron to the liver where it's stored, and that eventually makes its way by transfer into the bone marrow to be reused. And the heme ends up being transported via the bile or via the blood to the poop and the pee to be gotten rid of. Now, by having a higher red blood cell count, you have more oxygen carrying capacity, which means you can make more ATP for longer. Gives you endurance. And a lot of endurance athletes want to do this. Now, the natural way of doing this is to go to Colorado and work out at altitude where there's less partial pressure of oxygen, and you'll just naturally make more red blood cells. The more you exercise, the more you use oxygen, and you will make more red blood cells. So having a high hematocrit is useful in endurance athletics. Some athletes use, as a way of cheating, either injection of erythropoietin, EPO, which is a drug that's given to people, usually people with kidney disease, that need artificial erythropoietin, and this will raise their red blood count. Or the other thing that they do is they have a unit of blood or more taken from them, centrifuges down so the plasma is gotten rid of, and then they re-inject their own red blood cells into their body before competition, and they increase their hematocrit, and it improves their endurance. Lance Armstrong did this, and a lot of people in the Tour de France and people like that have been caught doing this. It's a very difficult thing to catch because it is their own erythrocytes. Basically, what they have to do is look at the hematocrit count before and then during and then after the competition to see if they're artificially elevated. The big danger is that it increases blood viscosity. And then the blood viscosity requires more blood pressure to move the blood because it's harder to move, which means the heart is working harder. It can damage the walls of the blood vessels. It can damage the heart. It can cause clotting and therefore cause strokes. But they'll lay the gold medal on the coffin if that's the case. It's really banned from athletic competition, but it illustrates how erythropoietin works and how red blood cells work. Anemia is the lack of this. This is more for the pathology course, so we're not going to get into it. But if you have defective bone marrow because of you get aplastic anemia. This could be radiation, that kind of thing. Hemolytic anemia is erythrocytes are destroyed prematurely. Erythroblastic, large number of immature cells because you have accelerated cell maturation. Hemorrhagic is because of blood loss, you hemorrhage. Pernicious anemia is not enough vitamin B12. And we'll talk about intrinsic factor later. Or you can have problems of the abnormal hemoglobin, it's genetic defects, that kind of thing. When we talk about erythrocytes, on the surface of each erythrocyte is an antigen. There's actually about 40 different types of antigens that are possible on the cell membrane. They're unique to red blood cells. We use this to type blood. Now, the self-antigens, these are proteins that will evoke an immune response, but not in yourself. In the one type of antigen is, for the ABO blood groups, is A. If you have that antigen, you are type A blood. If you have the type B antigen, you are type B blood. If you have both antigens, you are type AB because you have both. And if you have neither of the antigens, you have type O, which is actually type 0. Now, what's interesting is that people make antibodies against the antigens that they don't have. If they made antibodies, excuse me, against the antigens they do have, their immune system would attack their own blood. So they make antibodies against what they don't have. So type A people make anti-Bs. Type B people make anti-As. Type AB don't make any antibodies. And type O people make both anti-A and anti-B antibodies. So the anti-A react with the antigen A's, anti-B, blah, blah, blah. So you can't make the antibody against your own. So A has anti-B's, B's have anti-A's, AB's have neither, O's have both. And there's a little chart for this. So what this means is that if the person is type A and they're given a blood transfusion of type B blood, their anti-B antibodies will coagulate the B blood, and there will be problems. If they are given type AB blood, this anti-B antibodies will attack the B antigens on the AB blood, coagulate it, and there will be problems. If they're given type O blood, there are no antigens, so it accepts it. The antibodies have nothing to attack. So type A can get a transfusion from other A's or from O's. Type B, if they get an A, their anti-A antibodies will attack it. If they get AB, their anti-A antibodies will attack the A antigens on that. But O, no antigens, no reaction, so they can accept B or O. AB has no antibodies, so if they get an A transfusion, they're fine. If they get a B transfusion, they're fine. If they get another AB transfusion, they're fine. If they get an O transfusion, they're fine because they don't make any antibodies. Type O people make both antibodies. So if they get A's, B's, or AB's, the antibodies will attack the antigens on the donated blood. So they can only take from another type O. So type O, to summarize, can give to anybody, other O's, AB's, B's, or A's. Type AB's can take from anybody, but can only give to other AB's. B's can take from other B's or O's. A's can take from other A's or O's. So O is the universal donor, AB is the universal recipient. Now there are other antigens that are on the blood. The most common one is Rh, and it means rhesus monkeys, because that's where it was found. If you're Rh positive, you have this antigen. So if you don't, therefore, make antibodies against it. If you're Rh negative, you do make antibodies against it if you get exposed to Rh positive blood. It's like almost like getting an infection. So the first time you give somebody that's Rh negative blood a transfusion with Rh positive blood, there's no antibodies yet, and so the transfusion will work. The second time they're exposed, however, they have made the antibodies now, and it will get rejected. we just designate this as positives and negatives. So if you have all three antigens, you are AB positive. If you have no surface antigens, you are O negative. The difference with the Rh is that the antibodies are not pre-made. Now there's a number of other ones. There's something called Cal, there's Lewis, there's over 20 other types of antigens. They just don't play much of an effect because they're rare. So we call this agglutination if they get the wrong. It's what the antibodies do to the antigens. It makes them clump. So what we do to test your blood type is we put a drop of the reagent and see what reacts. If no clumping, it's a successful match. Clumping, it's a non-successful match. I will answer these questions on Tuesday if people ask. We will carry on with white blood cells in the next video.